everyone. My name is Anna Hermans.
And I'm Savannah Dale.
We both are graduate students in our second year in plant breeding and genetics at Cornell University, currently at Boyce Thompson Institute. We are 26 and 22 years old and have been studying biology for 8 and 5 years already. That sounds like quite a lot, but it's also so exciting and there's so much to discover.
Today we'd like you to join us in the behind the scenes of fruit crop development and plant genetics. When we talk about plant breeding and fruit crop development, we're talking about changing traits of plants to improve characteristics that we want, such as increasing the amount of food a single plant can produce or improving the flavor of that food. In order to improve traits in plants, plant breeders have to cross plants in the field and then determine which ones have those desired characteristics that they want. Plant breeders can use this pre-existing information to make decisions, but they can also use genetic editing in order to change these traits at the genetic level.
There are a lot of people involved in plant breeding. There usually is a scientific institution or a company who works on unraveling the genetics behind a specific species. Then there are farmers who are growing the plants in the fields for evaluation of how the crop grows or how much yield they can get from that plant. And then oftentimes there also is the community involved in the whole breeding project, because those are the people who will be eating the plant in the future.
As plant breeding students, Anna and I conduct research on food crops. Anna works on tomato in the Giovannoni lab, and I work on Physalis in the Van Ack and Gore labs. Physalis is the little cousin of the tomato and is an orphan crop.
So let's start by going to the greenhouse where I'm growing my tomato plants for research. We now are at one of Cornell's growth facilities, where several research groups are growing their plants under controlled conditions, which means controlled water access, controlled temperature, light, and also pest control. And here are my tomato plants. 95 2-month-old plants from all kinds of different tomato species. Today I want to show you my research based on 2 of those species. This tomato plant here is from the species called Solanum lycopersicum, which is the modern or cultivated tomato species. So most of the tomato fruits that you buy at the store come from this species, even though there are quite a few different varieties within this species. When you look at the plant, you can see it has quite a lot of leaf material, and the leaf nodes are serrated. This tomato species is called Solanum pimpinellifolium. It's a quite special plant because it's the wild ancestor of our domesticated modern Solanum lycopersicum. And you can see this plant looks quite different. The leaves are less, and they are also much more round compared to the other one. The stem is a little darker, and the plant overall looks a little thinner. Let's look at how the fruits of those 2 species look like. You can see that the fruit from the wild type Solanum pimpinellifolium species in the bottom is much smaller than the fruit from the modern Solanum lycopersicum species in the top, and the small fruit is filled with seeds. Large fruit size and high amounts of fruit flesh instead of seeds are traits that have been selected for during tomato breeding. But what exactly makes the plants from the 2 species and their fruits look so different? This must have something to do with the genetics. The genetic material of each organism is encoded in their genome, which basically is a long string of information about the organism called DNA, present in every single cell. This string can be partitioned into something called genes. Different combinations of genes play a role in different cells of an organism, which is why the cells don't all look the same. The information of these important genes is copied from DNA into mRNA through the process of transcription. mRNA is further processed into protein. To understand how the genetics differ between the 2 tomato species, specifically in the cells of their fruits, I need to look at the information encoded in their mRNA. The fruits that you just saw were cut into small pieces and frozen at -80 degrees Celsius. Pretty cold. To extract RNA from the cells, we have to grind the frozen material. And to do that, we're going to use a coffee grinder. So we're going to add our frozen material in there and a little bit of dry ice, which is the solid state form of carbon dioxide, which will help us keep our material frozen. Tomato snow. Some of the ground fruit material is transferred into small tubes in which the RNA extraction is going to take place. I have to cool everything down in liquid nitrogen, which is at negative 240 degrees Fahrenheit, to make sure my samples do not thaw. Different chemical solutions are added to break down the cell walls and to get the RNA into solution. The mix is spinned very fast in one of our centrifuges. Because some chemicals are water soluble and others are water insoluble, two layers will form, which have to be separated. RNA is in the upper water soluble layer. The liquid containing RNA is then sieved through a column, which acts like a microstrainer. The RNA sits on the column like pasta in a strainer, and the chemical solutions and cell waste are washed through. The RNA is rinsed with alcohol, and finally washed down through the sieve pores into a fresh tube with water. Those RNA samples will be sent to another lab for sequencing, which basically is reading the information that is encoded by the RNA, which is in the little solution here. The information, which at that point will be digital, will be sent to my computer inbox folder. The RNA information that I then get from the two different fruits can tell me about what genetic differences there are between the Solanum pimpinolifolium and the Solanum lycopersicum fruit. And those genetic differences must have to do something with how the phenotype, so how the fruit looks like. And this genetic variation is important to know for further breeding of improved crops.
In addition to studying the expression of DNA by measuring RNA like Anna just showed us, we sometimes want to change the DNA of an organism to better understand what a particular gene is doing. In my lab, we use CRISPR-Cas9 gene editing to do this. CRISPR technology allows us to cut DNA at the gene we are interested in using Cas9 nuclease, shown by the pink blob. When this DNA is cut, this allows us to either take out part of the DNA or put in a new piece of DNA. Then the cells naturally repair the DNA around our edit. We say the plants that we have changed the DNA in are edited. Removing a piece of DNA allows us to observe what happens in the plant when genes aren't functioning, whereas adding some DNA in allows us to see, for example, what happens when we have more of a particular gene expressed in the cell. This is the tissue culture room in the Van Eyck lab. This is where we grow our edited plants. When we first edit plantlets, they go onto petri dishes like this one. And as they grow larger, we move them onto magenta boxes like this one. Both the magenta box and the petri dish are filled with a gel-like substance called media, which provides nutrients to the plantlets. This is because the plantlets are so small and they don't have roots, so they can't take up nutrients from the soil on their own. These edited plantlets need to be transferred to new media every few weeks in order to provide them with a fresh supply of nutrients. This must be done in a sterile environment known as a laminar flow hood. Once they get large enough, like these plantlets here, we will transfer them into soil in the greenhouse where they can develop roots and grow into full-size plants. So this is the greenhouse for the Van Eck Lab, and I want to show you guys our gene-edited plants. This is what they look like when they have grown bigger. These have come from the tissue culture room, which we just saw. But here we have our plant, and it looks very similar, if not the same, as a non-edited plant. And this is because the gene that we've targeted only affects phenotypes that we can't see with our eyes. However, we want to make sure that we have the right genes in here that we targeted with our gene editing. So we're going to take a DNA sample. For our DNA samples, we are going to use tissue from a young leaf off of our plants in the greenhouse. The tissue will be placed in a sample tube to take back to the lab. First, in our DNA extraction process, we will suspend our samples in a solution to prepare them for the extraction process and make them easier to work with. Next, we need to grind the leaf tissue. This breaks apart the cells in the tissue so that the DNA is released from the nucleus. Next, we will use various solutions to clean the DNA and separate it from the other parts of the cell which we do not want. Finally, we can dissolve our DNA in water to make it easier to use. Now we are going to cut our DNA in specific places and make mini copies of those DNA fragments using a special PCR machine. After this, we can load our DNA pieces onto a jello-like agarose gel. We use an electrical current to push the DNA pieces through the gel and separate them by size. This is what the DNA pieces look like after this step. Now that we know we have the right DNA, we will send it off for sequencing, a process in which the DNA code is read. We can now use a computer program to compare the DNA sequence from our edited plants versus our unedited plants. This way, we can determine if we correctly edited our genes. Hopefully, if we did correctly edit our genes, we can produce a plant that has better characteristics such as nutritional quality or flavor. We have now shown you two different methods of targeting genetics in fruit crops.
In the first example, we looked at RNA variations in tomato.
In the second example, I showed you how we can change DNA in physalis. Both of these methods investigate genetic variation that can be used in plant breeding to improve food crops.
We hope you enjoyed the video.
Thanks for